This video is created to understand the basic features of dihybrid cross and their modification. So let's start. The first question is, what do you mean by dihybrid cross? So the term dihybrid cross can be differentiated into three small words. One is known as di. Here the di stands for two traits. Two means to say example is one is R and another one is capital Y. Capital R means it is the round and Y means it is yellow. So round is the seed shape and Y is the seed coat color. So we are differentiating into two traits. One is the shape of the seed, another one is the color of the seed coat. So two traits are involved here. That is why it is called di. Hybrid means each trait has two different alleles. For example, one is capital R, another one is small r. Here the capital R stands for round, but it is actually the dominant allele, and if it is smaller then it is the recessive allele. Simultaneously, if it is capital Y, then it is also dominant allele for the color yellow, and if it is smaller then it is the recessive allele for the color yellow. That means if an individual possesses only small y, they will be expressed green color. And now what do you mean by cross? Cross means there are two individuals, parental individuals, father and mother, which are combining or crossing their genetic information. So let us understand the dihybrid cross in more detail by taking a picture. So dihybrid cross is a breeding experiment between the parental generation organism that differs in two traits. The individuals in these types of crosses are homozygous for a specific trait, or they share only one trait. That is, here they are homozygous parental. One is capital Y, capital Y, capital R, capital R. That means purely yellow and round. And this one is small y, small y, smaller, smaller. That means it is basically green color and wrinkled in nature. So traits are characterized that are determined by the segments of DNA called the genes. So in dihybrid cross, parental organism have different pairs of allele. One pair of possess homozygous dominant allele. So this is the capital Y, capital R, that is homozygous dominant allele, and this one is the homozygous recessive allele. So the offspring in F1 generation or first filial generation, that will be all heterozygous for the specific trait being studied. This means that all of the F1 individual possess a hybrid genotype and express the dominant phenotype of each state, and that is why it expresses yellow and it is round, because they contains one capital Y and one capital R at least. So in this dihybrid cross, if we self-pollinate of these F1 generation plants, results in offspring. For example, this first filial generation plant can eventually produce four different gametes, that is capital Y capital R, capital Y smaller, small I capital R, and small I smaller. So these are the four different alleles, four different gametes actually, and the four different gametes are pollinated, and we can check the ratio. We can predict the ratio by using a Punnett square to reveal the possible outcomes of a genetic cross. For example, if capital Y capital R is mated with capital I capital R, then the product will be capital Y capital Y capital R capital R. So that means it is purely yellow and it is the round nature. So out of this 16 progeny, we can say there are 9 progeny, that are 9 seeds. About 9 out of 16 of F2 plants will have round and yellow, that is round and yellow, and about 3 out of 16 will have round and green, about 3 out of 16 will have yellow and wrinkle, and about 1 out of 16 will be green and wrinkle. So the green wrinkle means they will express all small. That means small y, smaller, smaller. That means all are recessive. That is why they will express green and wrinkle. If at least one capital Y or capital R is present, they will express that capital Y segment or capital R segment. For example, in this particular case, this is the gametes, that is small y capital R and small y capital R once again. So the product will be small y capital R, because all of them are y, small y, that means they are recessive, so the expression will be seed coat will be green color. But because it contains both capital R or at least one capital R if present, for example in this case at least one capital R is present, so they will express themselves as round. But in this case no capital R is present, in this case no capital R is present, that means they will be wrinkled in nature. So the phenotypic ratio will be 9:3:3:1. But this is not actually true for all the cases. For example, this is the modification of dihybrid ratio or interaction of genes. In a dihybrid cross, one does not always get the standard ratio of 9:3:3:1. Two non-allelic genes may interact and modify the expression of the offspring. For example, in this example, in sweet pea, two white flowered varieties were crossed. So these are the white flower varieties. So this is also white and this is also white, and these two white flower variety have crossed, and the progeny is colored, that is purple colored. In the F2 generation, the ratio was 9 colored and 7 white. This is because one of the white flower plants had the gene for color C, that is the capital C. This one capital C for the gene color C, but no gene for the enzyme to make the color visible. That means it is small p. So they had a gene for the color C, but no gene for the enzyme to make this color visible. In the other case, white flower parent had one gene for the expression of the enzyme capital P, but no gene for the color production. So the F1 had both the genes, capital C and capital P, that is a gene for color, that is C, and a gene necessary for the enzyme, that is capital P. So they had colored flowers. So this chart explains that out of these 16, 9 had colored because they had at least one gene for the color capital C and one gene for the enzyme P. So we can explain this thing, that is how the 9 came from. Number 1 at least capital C and capital P, and this one number 2, number 3 capital C capital P, number 4 also there, number 5, but not number 6, because this number 6 contains both capital C but both small p, that means they will be white. So the purple colored product will be at least one capital C and at least one capital P. In this case number 7, one capital C and one capital P is present, that is why it is called purple. So we can count this thing total, and we can count that they are actually 1 2 3 4 5 6 7 8 9. These are the 9 had colored because they had at least one gene for the capital C color and one gene for the enzyme capital P color. 7 had either the gene for color or the gene for the enzyme, but not together, so they will be all white flowers. So the ratio is 9:7. Hope you understand.